So today we'll be covering second chapter, and that's chapter on terpenes. Rather large one and a little bit challenging. Not very challenging, but a little bit challenging. Also, to let you know about quizzes and exams. Quiz is of course today, but you still have some time after this class to study a little bit and then take a quiz, because a quiz is 15 minutes long, closes at 8, and actually gave you 5 minutes past 8 in case there are some problems. So you can start as late as 7:45. And since we'll be finishing this by about 4:30, if you don't have any other classes, you can study for a couple of hours before taking a quiz. The purpose of the quiz is to check basically whether you are studying, and to encourage you to studying and to study these PDFs that I'm posting, basically textbook, what is our textbook. So the only thing that you need for the quiz is textbook. Read the textbook, and if you read it, learn the material, are familiar with the material, you should do well on the quiz. For the test, you may need a little bit more, and I have been posting some videos. Unfortunately, quality is actually rather poor. I don't know what is the problem, but when I'm now making videos, I'm being forced to update the old files. Simply software is asking me, do you want to update? Because without it, it won't do it. And when I update them, video or audio quality becomes really poor. And without update, there is no audio. And so I don't have time to re-record the audio. So hopefully that's not going to be too much of a problem. And also these videos right now are not really that important. If you watch them, that's all right, but they're not really that important. And maybe I'll be able to fix that for the future videos. So now when we consider terpenes, they are secondary. Oh, just a minute. I guess a little bit fighting again. So terpenes are a rather large class of secondary metabolites, and they are kind of typical secondary metabolites. They are produced by individual species. They are not common. Very few terpenes would be, or actually I don't know of terpenes that are common to all the living organisms, and very large number of them are present, and they play various roles. So they could be protective roles like antifeedants, insecticides, bactericides, fungicides. They may play a role in communication between different individuals of the same species, like pheromones, or within the same species between different tissues and organs, like hormones. And a lot of terpenes actually have important pharmacological activity. And so they are either themselves medications or starting points for developing different medications. Traditional name for this class of compounds is terpenes. But more recently, they are being called terpenoids. And the reason for it is that there is a view that suffix "-ine" should be reserved for things like alkenes. And so we should not calling it terpenes, but at the same time, we should be calling these compounds terpenoids because other natural product classes, such as alkaloids, steroids, icosanoids, have this "oid" ending. So both are in use. Formally, terpenes are derived from isoprene, and this is formula of isoprene. So 2-methyl 1,3-butadiene. But formally, that means they're not actually derived from isoprene. They are actually derived from isopentyl pyrophosphate. So that's the actual starting material. And in this skeleton, we can identify tail, head, and tail. So head is that part where methyl group is. So this would be head, carbon 1, and tail would be carbon 4. Typically, individual isoprene units connect head to tail, and then form terpene molecules. Alternatively, there is tail-to-tail bonding. And finally, there could be irregular terpene skeletons where we cannot identify head-to-tail or tail-to-tail. Irregular terpenes are generally restricted to monoterpenes. Just stop sharing. Okay, so it is important for you to be able to classify terpenes. And let's look at classification. So you have to know how to classify terpenes. They are classified according to number of isoprene units. And since each isoprene unit has 5 carbons, then in turn, actually terpenes are classified according to number of carbon atoms. So one isoprene unit or 5 carbons, that's hemiterpene. That means half terpene. The reason for it is that originally when terpenes were discovered, they had 10 carbon atoms. Those originally discovered, the smallest terpenes had 10 carbon atoms. And so they were named monoterpenes because their biosynthesis was not known. So they were named monoterpenes, and that's 10 carbons or 2 isoprene units. And so now when those with 5 carbons were discovered, they had to be named somehow. And so they were named hemiterpenes, and that means half terpenes. Then we have sesquiterpenes, that's 1.5, that means 3 units or 15 carbons. Diterpenes, 4 isoprene units or 20 carbons. And the next one, be very careful, you need to know correct spelling. They are sester terpenes. So sester terpenes, not sesterpenes, but sester terpenes. Triterpenes, 6 units, 30 carbons, and then new class that was discovered about 10 years ago and named even less than 10 years ago. They are sesquiar terpenes, so sesquiar terpenes. 7 units or 35 carbons. I think only about 20 of those are known, and tetraterpenes. I think there may even be some larger known, but those are very rare, like 1 or 2 compounds of each or something like that. So besides classification, you have to know biosynthesis. And so here is first, I won't be going in detail. I'll point only some aspects because most of this chapter is biosynthesis, and to do it in detail, it would take several hours. It would take several lectures like this. So biosynthesis starts with synthesis of mevalonic acid. And so for that reason, this pathway, this biochemical pathway, is called mevalonic pathway or mevalonate pathway. And that pathway is separate from acetate pathway. So even though mevalonic acid, which is this one middle, is synthesized from acetyl coenzyme A, and acetyl coenzyme A is typical of acetate pathway, this is separate biochemical pathway. It is not acetate pathway, it is mevalonate pathway. So 3 molecules of acetyl coenzyme A combine to give mevalonic acid, and then decarboxylation and phosphorylation results in formation of isopentyl pyrophosphate. This is OPP pyrophosphate group. In biochemistry, we typically call it pyrophosphate. In chemistry of natural products, it's usually called diphosphate, not pyrophosphate, but diphosphate. Both terms are used interchangeably, and so I use them at random in this chapter, pyrophosphate or diphosphate, so that you get used to both terms and know that both terms mean the same thing. So starting material for biosynthesis of all terpenes is isopentyl pyrophosphate. And here is how it works. Isopentyl pyrophosphate actually isomerizes, well, I have it later. Isomerizes into more stable isomer called dimethyl allyl pyrophosphate. And dimethyl allyl pyrophosphate gives rise to various hemiterpenes. As I said, there are not many of them. Dimethyl allyl pyrophosphate dimerizes with isopentyl pyrophosphate, or diphosphate in each case, to give geranyl pyrophosphate. And geranyl pyrophosphate is precursor of monoterpenes. Condensation of geranyl pyrophosphate with another molecule of isopentyl pyrophosphate gives farnesyl pyrophosphate, and that's precursor of sesquiterpenes. Two molecules of farnesyl pyrophosphate, when they combine tail to tail, give triterpenes, and modified triterpenes are steroids, covered in the next chapter. That will be on the first test in 2 weeks, but not on the quiz today. Steroids are not in the quiz. Triterpenes are. So then, alternatively, here obviously biosynthetic pathways branch, a reaction of farnesyl pyrophosphate with isopentyl pyrophosphate, so yet another molecule. You can see each class of terpenes is obtained by polymerization, adding another unit of isopentyl pyrophosphate. So we get geranyl pyrophosphate. That's precursor of diterpenes. Dimerization of geranyl pyrophosphate gives tetraterpenes or carotenoids. Tail to tail, they are carotenoids. I believe that there are a few head to tail, and then those would be typical tetraterpenes, and they would be originating from different compounds. Basically, it would be like octaperennial pyrophosphate. You don't need to know that. 8 perennial units. And by the way, this isopentyl unit is also called perennial unit. And so that's another synonym. A lot of synonyms here. Finally, geranyl pyrophosphate reacts with isopentyl pyrophosphate to give geranyl farnesyl pyrophosphate, and that's precursor of sester terpenes. So that's a summary. And then here we are looking in details. As I said, I won't be going into details, but here you have detailed description of how mevalonic acid is formed. So it's typical Claisen condensation of acetyl coenzyme A to give acetoacetyl coenzyme A, and then a reaction with another molecule gives this hydroxymethyl glutaryl coenzyme A. And then reductase reduces one of the hydroxyl groups. Hydrolysis, which means loss of acetyl coenzyme, or actually loss of coenzyme A bond to sulfur, results in formation of corresponding aldehyde, another reduction, mevalonic acid. And then mevalonic acid is decarboxylated. It loses carbon dioxide. 3 molecules of ATP are involved in this to give isopentyl pyrophosphate. Isopentyl pyrophosphate with catalyzed by enzyme, isomerase enzyme, isomerizes to more stable dimethyl allyl pyrophosphate. But both are needed for biosynthesis of terpenes. And note that all of these reactions are stereospecific. In all of these reactions, when hydrogen is lost, it's pro-R hydrogen that is lost. And biosynthesis of terpenes, including monoterpenes, are perennial transfer reactions. That means transfer of isopentyl group or perennial group. And reaction is then appropriately catalyzed by enzyme appropriately named perennial transferase. So dimethyl allyl pyrophosphate loses pyrophosphate or diphosphate group to give this very stable cation, dimethyl allyl cation, and then that cation in electrophilic addition adds to isopentyl pyrophosphate. You can expect this from organic chemistry, that cation would add to double bond to less substitute carbon, Markovnikov rule, because this gives more stable tertiary carbocation. And again, loss of the same hydrogen. It's always same hydrogen that is lost, pro-R hydrogen. And it's because enzymes catalyze it, and they catalyze, always is the same type of enzyme that catalyzes removal of the same type of hydrogen to give double bond. And so that's geranyl pyrophosphate, precursor of monoterpenes. And then from there, this is kind of schematic representation how various monoterpenes are formed. Because we really spent entire course on terpenes, so it's highly abbreviated. I had to make a lot of choices, and so even classification of monoterpenes is not something that we are looking at in detail. although we'll cover monoterpenes with more detail later. Okay, at this point, actually, let me stop. First, let me see. Yeah, okay, well, there's one more. She's joined us. So, are there any questions? If you do have questions, turn your sound on and ask a question, or you can type it up. Let me just check. Actually, I'm not looking at chat. So let me see. Hey, oh, where is chat? There it is. Okay, yeah, nothing. Okay. And so sesquiterpenes biosynthesis proceeds the same way, simply polymerization with another molecule of isopentyl pyrophosphate. So now it's geranyl pyrophosphate, it loses pyrophosphate group, cation, and so on. Now, sesquiterpenes are the largest group of terpenes. There are the most of them of all terpenes, and there are actually 200 classes, different classes of sesquiterpenes. So there are tens of thousands, I believe over 50,000 different sesquiterpenes. And most of them are a result of cyclization. And so here is, I don't expect you to really memorize this. I just want to show you how cyclizations work. And this is typical cyclization to form humulene as intermediate, and then from there, humulene could cyclize in different ways to give different compounds, very different compounds. And so here is one to give rather complex compound, pentylene. So 3 fused 5-membered rings. And then diterpenes, sesquiterpenes, biosynthesis proceeds the same way. So just extension of previous procedures. And again, cyclizations, you can guess how they work. Although again, you can see in this example of retigeranic acid, how enzymes will hold the molecule in particular conformation that is suitable to close particular rings, in this case, particular sequence of 5 and 6-membered rings. And obviously different enzyme will fold the chain differently and different rings will close. And that's why we have so many different classes of sesquiterpenes, diterpenes. Now, triterpenes and tetraterpenes are different from rest of the terpenes because they are biosynthesized from 2 identical units in tail-to-tail fashion. Triterpenes from 2 farnesyl phosphates and tetraterpenes from 2 geranyl pyrophosphates or diphosphates. Of course, when pyrophosphate or diphosphate group leaves, you end up with cation. And if from 2 groups, 2 pyrophosphate were to leave, you would have 2 cations. And you cannot have 2 cations combining with each other because they're positively charged. So mechanism obviously has to be different. So allylic cation of 1 farnesyl pyrophosphate, so it's farnesyl cation, adds actually to double bond, electrophilic addition, and again to less substitute carbon to give tertiary carbocation. And then that results to formation of intermediate cyclopropyl ring, which undergoes ring enlargement to cyclobutyl group, and then ring opening of cyclobutyl group gives new carbocation, which is then reduced by NADPH to give basic hydrocarbon that is a precursor of all of the triterpenes, and that's squalene. Very important compound, squalene, so you need to know it. In case of tetraterpenes, reaction is analogous to an extent. So geranyl pyrophosphate loses pyrophosphate group, adds to another molecule of geranyl pyrophosphate. Cyclopropyl intermediate forms, undergoes ring enlargement to cyclobutyl. But then when cyclobutyl ring opens, it doesn't undergo reduction. Instead, it undergoes elimination of hydrogen to give additional double bond. And that results in completely different class of compounds with completely different properties. from triterpenes. And so while triterpenes, squalene, was precursor of steroids, these are, because of the middle double bond and restricted rotation, ring cannot easily fold to make additional rings. The only rings that could be formed are here at the ends of the chain, because that's not affected by this middle double bond. But those complex polycyclic systems cannot form. So we end up with a class of compounds known as carotenoids. So those are details of biosynthesis. And now rest is just description of different classes of terpenes. Before I do that, again, I want to see if there are any questions. How far will quiz? It covers entire chapter 2, but there isn't really, I don't know how to put it, there isn't really that much, it's not, I don't think it's very challenging. A test may be more challenging, but quiz, I don't think will be that challenging. But yeah, you need to know the entire chapter 2. As I mentioned before, for those who joined the class early, you can take a couple of hours, let's say from 4:30, actually even 3 hours, from 4:30 to 7:30 and study. And test is mainly on whether you read the material. So here are simply examples of hemiterpenes. So you can see there are very few of them. Monoterpenes are much more numerous. So they're open chain monoterpenes, and you can see how they are formed. And then a closed chain. And so you can see, or cyclic. Actually, these are still open chain. So the open chain, all of the monoterpenes usually have very nice fragrance. And so they're used as fragrance and flavor compounds. And some of them have some other use, but that's their main use. Like they could be used as disinfectants. And then here are cyclic. So this is one typical ring, monocyclic. This is called pimentane type ring. And various classes, even aromatic. These are all regular, normal rings. So limonene has this pimentane type. All of these have pimentane type. Even when another heterocyclic ring forms, like furane. Now, when menthil ring, this menthil ring, pimentane type ring, actually forms cation. Cation is really formed from limonene by addition of proton. That's menthil cation. Now, this menthil cation can cyclize. Double bond donates electron pair to cyclize, and depending which carbon of the double bond forms a bond. Red would be this carbon right here, carbon 1. Blue, this carbon, carbon 2. We get either bond, we obtain either bornyl cation, which then, depending on biochemical pathway, could give borneol and camphor, or camphene. And notice camphor and camphene have different skeleton. Or if this carbon forms a bond, then different bicyclic ring is formed pineal cation, which gives rise to alpha and beta pineins, obviously from pine oil, or as a result of rearrangement, fenchil cation, and then fenchil and fenchil. You have to be able to recognize these compounds when you see them, so you should be familiar with different types of drawings like this. Menthil cation can undergo additional rearrangements to gamma, it could actually lose proton to give gamma terpenin, which would be a simple thing, or form three-membered rings, like tuggyl cation, goes all the way to tugron, or delta 3 carine. And you can see tugron has 5 and 3-membered ring, while carine 6 and 3-membered ring. So you can see wide variety. And then there are iridoids, which contain cyclopentane rings. So in this case, cyclopentane ring is formed. They're still regular skeletons. And important iridoid is nepetalactone. That's actually catnip, compound that makes catnip smell like catnip. And so that's cat attractant. And then there are those that are irregular. So you have to be able to distinguish regular from irregular. So irregular are not head to tail, and then as a result of formation of geranyl pyrophosphate, then various cyclizations, rearrangements, and so on. In this case, actually, as a result of reaction of dimethyl allyl cation, basically what happens is dimethyl allyl pyrophosphate does not react with isopentyl pyrophosphate. Instead, reacts with another molecule of dimethyl allyl pyrophosphate. So dimethyl allyl cation reacts with another dimethyl allyl pyrophosphate, and at the end, cyclopropyl ring forms. And so cyclopropyl ring means irregular skeleton. And then, so those are all irregular skeletons. And then any opening of the cyclopropyl ring will also result in formation of irregular skeleton. So if you see any monoterpene that has this skeleton, that's irregular, or any of these rings, and it opens there also. So there are 4 types of skeleta that are irregular. So make sure to know what is irregular skeleton, because often students may think, oh, something like this must be irregular because this is so complex and this is complex, and even these 5-membered rings. But no, they're all regular skeletons. And they're regular because they are a result of biochemical pathway that originated with geranyl diphosphate. But this is not geranyl diphosphate. This is 2 molecules of dimethyl allyl pyrophosphate that condensed. And it's very similar to what we have seen in the way triterpenes and tetraterpenes are made. When you have 2 identical molecules condensing and then forming that intermediate cyclopropyl cation and then opening and so on. So that's what it's analogous to, and that's why they are irregular. So that's a very important point. Since I made this important point, are there any questions? Okay. And then sesquiterpenes, again, just illustration of large diversity of sesquiterpenes. There are so many of them. So they originate from farnesyl pyrophosphate. So there are quite a few of them. And they have also very beautiful fragrances, even nicer, even more beautiful, more pleasant fragrance than monoterpenes. So they're used a lot in perfumery, either themselves or as constituents or various oils, natural oils. And some of them are even cheap fragrances. We'll cover them later. I think farnesol is used as a fragrance in those fabric softeners, which is not very good because similar compound attracts fire ants, which are common here in South Florida. So if they get to smell it, they could mistake it. It's actually not fire ants attractant. It's similar to it. But what happens is it attracts, just a few of them get confused and get attracted by it. But if that happens, they will leave their trail, that they have been attracted by something. And now what they leave, the trail that they leave is actual trail. They are sending a chemical signal to other fire ants. We are following an attractant. And now other ants won't be confused because they are following the actual trail. And in no time in your freshly washed clothing, you have tons of fire ants. And the problem with fire ants is they don't just sting right away if you have ever been stung. They actually first swarm before they start stinging. And so you may not realize that there is a problem until you get multiple stings by fire ants. It's not like one is going to sting you and realize, okay, there is one, and now I can stop. They are actually really sneaky in that way. And then farnesyl cation, of course, can cyclize. It can cyclize to give various cations. We have seen humulyl cation before, which is the 11-membered ring. And then it rearranges to caryophyllene, another compound with rather nice fragrance. Or 10-membered germacryl cation, and so on. And then all various rearrangements. And now when you get compounds like this that are highly functionalized, they usually have various biological activities, often like insecticide, bactericide, antifeedant, and often they are of interest to us because they often have also pharmaceutical activity. And so they themselves may be interesting as medications or as a lead to develop a medication. But in this case, it's mainly just for you to, you cannot possibly memorize all these things or even recognize them when you see them. It's more just to understand the process. So if you're given something on the test, you can explain it or at least rationalize it and show that you understand it. And also that you know what is going on, basically, and be able to appreciate variety of different structures. If my cat doesn't stop, I'll have to lock her up. And here you can see uh, various diquinanes. Quinane means five-membered ring. And so diquinane means two fused five-membered rings. And these are all classified as diquinanes because they have two fused five-membered rings. And the fact there are additional rings doesn't count. And then when you have three fused five-membered rings, they are triquinanes. And you can see variety. They could be linearly fused. This is angular. Something like this is on your extra credit question. And by the way, all of them should have three five-membered rings that are fused angularly, and then they should have four carbon atoms sticking out. So they could be four carbon atoms, like CH3 groups, or there could be functionalized, like with OH, or aldehyde, CHO, or carboxylic acid. All of that is correct. None of that is wrong. So four carbons sticking out, or two of them being on the same carbon atom, none of that is wrong. So when you're solving which structure is not correct, the fact that four of them are substituents, that's normal. And all of them do have four, you see, 1, 2, 3, 4. And this is modafinil propylene ring, so interesting, 1, 2, 3, 4. When making models, these in the plane of the drawing obviously should be in the same plane, but this one is down. So you put that one down on the tetrahedral carbon. Now, if this one is down, actually you would put these two up, if this is in the same plane. This is up, you would put these two down. I didn't represent this one, but in this one, this actually, you see this, I just noticed that I did not represent this stereochemical center. Here it's represented, so I can show you on this. And here you have both up, both down. So you see these centers are represented. This has to be represented. I did not show it here on isocomin. So you see, you would put down, down. That means these two up, this is up. And pentylene, I didn't show this. I should have actually. This one is a good example. See, down, down, and this one is strong down. These are up, 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 and so on. So when making, again, down, down, up. Or alternatively, we could represent an enantiomer, which means this would be up, up, down, up, and so on. A representation of an enantiomer is also fine. And if you don't know absolute configuration, we know compound is chiral. There are two enantiomers. We simply at random arbitrarily represent one enantiomer. And so some authors will represent one enantiomer, some will represent opposite enantiomer. And both representations are correct. So the fact that you may see in another paper same compound as enantiomer, that's not wrong. That's not. So to solve the problem, what I would suggest is make a model. Make a model of each of these skeleton, make it correctly, and if you make each model correctly, you should be able to solve that problem. And then diterpenes, when we get to diterpenes, even though there are lots of them, really a lot, diversity begins to a little bit decrease. So there are a little bit fewer of them compared to sesquiterpenes. And one of the very important skeleton is this taxadiene. It's basic skeleton of Taxol, which is Taxol is a compound that is used quite a bit in treatment of cancer. It's not a cure, but it does prolong life. And then there are some other important diterpenes. Another one is steviol. I would advise you against consuming this, but it's used as artificial sweetener when it's attached to various carbohydrate groups because it's very sweet. It may have other, it's diterpene, so it may have other biological activity and not very good idea to consume it. And then by the time we get to sester terpenes, 25 carbons, number of compounds decreases quite a bit. So now we are encountering relatively small class of terpenes, but they're still present in quite a bit of organisms. And because they're such a small class, they were actually discovered relatively recently, 1965, which for terpenes is recent because original terpenes, first ones were discovered in 19th century. But still we have several hundred of them. So you can appreciate there, you may think, well, several hundred, that's a lot. Yes, it's a lot, but compared to like, let's say, close to like 50,000, I think, of, actually 50,000 maybe total number of terpenes, but probably like 30,000 of them are sesquiterpenes, then that's very small class. Some of the best known are something like retigeranic acid and scalarin, which has this typical scalarin skeleton of 4 fused 6-membered rings. And then triterpenes, very important class, but they are important not because of themselves, but because they're precursor for steroids. And I had to fix this, so this part won't be on the test because I made mistake. This is R1, so make sure to download R1, revision 1 of the chapter on terpenes, because I made a mistake with biosynthesis of protostereocation. So protostereocation gives rise to lanosterol, which is precursor of cholesterol and other steroids. Cycloartenol, precursor of plant sterols, and this is curbataxin. I'm not familiar with it, but obviously it's an antibiotic. Alternative mode of cyclization gives the marenyl cation, and then that gives other compounds, other triterpenes. We will cover azadirachtin, very important plant antefeedant or insect antefeedant produced by plants. Quasin was another compound that was very popular in the 90s because it's synthetic challenge. It's difficult to make in the lab. So a lot of labs were working on it and successfully synthesized it. Sesquiterpenes are very new class. They have been just discovered like in past 10 years, about 10 years ago. Actually maybe 9 years ago. Well, between 9 and 10 years ago, I believe in 2012. And then they have been, when they were discovered, there wasn't even a name for this class of terpenes. So about 3 years later, I believe they were named sesquiterpenes. So very new. And this is what they look like. They are head to tail compounds. So they are not tail to tail, but head to tail. So heptaprenyl diphosphate is. So 7 isoprene units in normal way, head to tail. So basically you start with dimethyl allyl pyrophosphate, and then another isopentyl pyrophosphate adds, and another, and another, and another, until you end up with heptaprenyl. And then cyclization, and various cyclizations give various compounds. And then tetraterpenes are pretty much all of them carotenoids. They're used interchangeably. There are some actual tetraterpenes, but they're very rare. And so carotenoids are very important. They are pigments, and they're edible pigments because we eat lots of them. And that's what their importance is. They're considered to be protective chemicals. They protect against oxidative damage, so they have pretty important health benefits, and it's good for us to eat lots of them if possible. There are also precursors for vitamin A, or retinol, and this is actually diterpene, but it's not formed in biosynthetic pathway for diterpenes. Instead, it's formed by cleavage of the middle double bond of tetraterpenes. And some of them are actually formed by cleavage, like cleavage of individual units, few carbons at a time until half of the molecule is cleaved. But either way, so vitamin A, even though you can identify it as diterpene, is actually not a diterpene. It's a product of degradation of tetraterpenes. Now, there is at least one known head-to-tail tetraterpene, so it would be real tetraterpene, and that would be this longestine compound. And finally, there are other terpenoid natural products. Those are terpenes. Those are products that have terpene side chain attached to something else. So, like aromatic compounds or something like that. Something that originates from, for example, shikimate pathway or acetate pathway. And so, vitamin E or alpha tocopherol is an example like that, where phytyl pyrophosphate attaches to homogenistic acid and then cyclizes to give chromane ring. So this is actually diterpene unit. And that also shows dangers of trying to identify terpene by counting. Now on the test you may not have a choice. When I give you a molecule and ask you what kind of terpene it is, you have to count isoprene units or carbon atoms. But here if you count, you see you have 4 original isoprene units. And actually you can find another one right here. You can find 5th and possibly even 6th. And you could say, well, this is triterpene. Actually, it's not. This comes from homogenistic acid, these 10 carbons, and that has nothing to do with, well, actually, yeah. You see additional carbons are added later, this carbon, this carbon. So these 10 carbons come from homogenistic acid, additional carbons that are added, probably from S-methylmethionine, something like that. So this is actually diterpene unit, not triterpene. But in this case, you would actually have to memorize. You would have to be told that. You cannot reason it out on your own. Well, for other terpenes, like, well, not a good example, like, okay, this one, you could actually say it's tetraterpene simply by counting individual isoprene units, like here. And even here, you can identify those units. Sometimes you have rearrangement, and so units are difficult to identify. Vitamin K also has diterpene side chain, and various menaquinones have chain of different number of isoprene units. Coenzyme Q is basic skeleton. Coenzyme Q0 is ubiquinone. It has no terpene side chain, but in living organisms, for it to function as antioxidant, it needs this terpene side chain. So CoQ6 is typical one in bacteria and CoQ10 in animals like mammals. And finally, tetrahydrocannabinol. Tetrahydrocannabinol is a terpene, and it's a monoterpene that is attached to a phenolic ring. That's the active principle of cannabis. That's what makes people high. Although people who took this actually don't like it because apparently there are other compounds in cannabis, and so it's a combination of all of them. That's what people like, not this individual compound. Because while this compound gets people high, it also causes very serious feeling of anxiety, and people really don't like that. And so here we can identify in delta 9 tetrahydrocannabinol actually 3 different structural elements. Monoterpene unit, you see 2 terpene units shown in bold, 2 isoprene units shown in bold, phenolic unit, and then this 5 carbon side chain. This would be kind of probably from acetate pathway. Okay. And so that completes our review of chapter 2. And I can see one question. Traquinanes could only have 4 carbon substituents sticking out. That's correct. Because they have 15 carbons. 11 carbons are used up to make 3 5-membered rings. When 3 5-membered rings are fused, that requires 11 carbons. And since there are 15 carbons, 4 are left, and so they could have only 4 sticking out, or 4 substituents on the rings. Yeah. Okay, any other questions? Okay, I'm going to stop recording now, and if recording turns out to be all right, I will post it.